Good early afternoon, everyone, and thank you for joining us. As Jill just said, I'm the director of the Biospecimen Collection and Banking Corps at the Simon Cancer Center. It's my pleasure today to introduce one of our key team members, and that is Emily Nelson, who is the Regulatory and Data Manager for BC Squared, as we've shortened that incredibly cumbersome name to. So in that role, she not only worries about keeping our noses clean from a regulatory standpoint, but also has a crucial role in everything to do with data, including which tools we use, where the data sits, who has access to the data, looking forward to new tools. She basically translates everything that the information services people say in their own language to a language I can actually understand. She has been with ORIEN, the Oncology Research Information Exchange, from the very start at IU. This is, to give you a perspective, the BC Squared has only really been formally in existence for 2 years, but she actually was the manager for ORIEN and the key person there since its very start. It has had a huge role and a lot of participation from our precision genomics program. So I will let Emily take it from here and explain what ORIEN does and what an incredible resource it is for most of the people on this call. Go ahead, Emily.
Thank you, Anna Maria, for that lovely introduction. It's such a privilege to have the opportunity today to come and speak with you about ORIEN, the Oncology Research Information Exchange Network, and the banking services that we offer through the Biospecimen Collection and Banking Corps at IU Simon Cancer Center. One of the things that we as an institution are really committed to doing is finding new ways to better treat, diagnose, hopefully prevent, and ultimately cure cancer. And this is one of the tools that we felt would be a really amazing opportunity to do that. So it's such a privilege to share that with you today. In 2018, IU Simon Cancer Center joined this consortium of cancer centers in the Oncology Research Information Exchange Network, or ORIEN, with that goal in mind of accelerating cancer discovery and delivering hope through collaborative learning and partnership. Here on this map, you can see the 18 different cancer centers that are currently a part of this growing network. And this consortium is managed through a company who recently went through a name change and is now Aster Insights. The majority of these cancer centers are all NCI designated, but there are 2 community cancer centers in here as well. The way that as a consortium, we work together is through this shared protocol. It's called the Total Cancer Care Protocol. And it's through this protocol that we engage our patients in a partnership to build a better future for cancer care. The amazing thing about having this shared protocol is that it allows us as a consortium to share information and data about our enrolled and consented participants. So here at IU, we have around 20,000 participants that have been enrolled through either some of our legacy banking protocols or through active consent to this protocol. And then across the consortium, there's about 325,000 participants that have agreed to engage in this. So when our participants sign up, they agree to be followed for a lifetime, allowing us to review their clinical data for important clinical follow-up information. They agree to donate biospecimens both prospectively and retrospectively. So if somebody is late in their cancer treatment, we have the ability to access archival biospecimens that are in pathology departments. We tend to be very restricted in accessing those archival clinical care materials because it is critically important to us to protect the care of our patients. So we often do not access those materials until a time that our patient is no longer with us. Those individuals who consent to participate also agree that they will are open to being contacted for future research opportunities. That can happen in a variety of ways. We've had researchers come to us and say that they're interested in connecting with a specific cohort of patients to learn more about their engagement in research. So we worked with a group of underrepresented patients to engage them and allow them to learn about this opportunity for additional research. There's the potential that these patients may also be connected with clinical trials that they are identified for as a result of their participation in total cancer care. And they also allow, as I already mentioned, for their data to be shared across multiple stakeholders. One of the projects within total cancer care and this ORIEN consortium that is especially appealing to investigators is the ORIEN Avatar project. And ORIEN Avatar is a marrying of deep longitudinal clinical data and molecular data from TCC consented patients. So one of the challenges of real world data is often that the data isn't structured consistently. It is difficult to manage. It's very unwieldy. And it takes a lot of human effort to organize that information in a way that it can be effectively analyzed. You know, in my conversations with biostatisticians, about 80% of the work isn't doing the analysis, it's just organizing the data in a way that you can use it well. So one of the beautiful things about this is that clinical data is really heavily structured. It is then paired with a germline sample and a tumor sample from that given patient. And those samples are shipped out for sequencing, for genomic sequencing. There's a whole exome for the germline sample, a whole exome, and a transcriptome for the tumor sample. All of this information is married together and made of. The way that patients are selected for Avatar has changed over time. But the intent has remained relatively stable, which is to help facilitate future cancer research in groups of patients with the highest need for better treatments, better care. You can see here on this chart that there are many different types of cancers that have been included in this ORIEN Avatar project. So on the left-hand side, in that black number, are the number of participants within the consortium. And then on the right-hand side are the number of participants that have been locally included in the Avatar project. And I think this is a really important distinction to make because within the consortium, there is the theoretical notion that we can procure samples across institutions and aggregate those for one investigator. And it has remained in the theoretical for as long as I have participated in this collaboration. So the guarantee with these on the left-hand side is that this information, once the request process has gone through, it would be available for use in research setting in a dry lab. And those instances in blue on the right-hand side are cases from our institution where there is the potential that we will have additional biospecimens banked within our lab and we can facilitate cancer research in a wet lab setting. As I mentioned earlier, our clinical data is extremely structured, and here on the right-hand side are the 18 clinical domains. So we capture information about the patient's diagnosis and family history for first-degree relatives, any instance of either regional or distant metastatic disease, any medication that they've received for their cancer care, and so forth. With this imaging tab, it's really important to know that that is in a flat format that is not the actual images that you would review on imaging software. So across the network, across those 18 different institutions, that avatar data is aggregated, cared for, and stored centrally with Aster Insights. And they make it available for investigators to use in order to develop initial cohorts and to query and kind of play around with the data. There is the ability to access that through a closed instance of CBio portal that's hosted by Aster Insights. So if you should have any interest in evaluating that data in that space, please feel free to reach out to myself or to our ORIEN program manager, Heather Penwell. And our contact information is on the screen for you now. As I mentioned, this consortium was really developed to facilitate collaborative learning and support investigation across the institutions. The ability to have, earlier on, a few slides earlier, I showed you the number of avatar participants that are currently available across the consortium. And it's just under 25,000 individuals that have that annotated longitudinal clinical data and omics matched. It's important to know that that is a larger volume of patients than what is currently available in TCGA, which has longstanding been a critical resource for the advancement in cancer. And different from TCGA, we have a mix of patients in their disease phase, and we also have this really beautiful longitudinal clinical data, whereas TCGA has a really solid SNP, and that's it. So with this information, we really want to foster engagement with our investigators and our colleagues. And as an institution, we are extremely committed to honoring the commitment that we've made to our patients, that we are going to use their generous gift to facilitate future cancer research. So our institutional policy is that we support any project that asks to use our data. We feel really strongly that that is how we can best engage in this and honor that commitment. So here we are highlighted in the yellow. Even though we are the youngest member of the consortium, we are the number 5 contributor to ORIEN intermember projects. And as you can see, we've led a few different intermember projects, but we are always looking for new opportunities to support our researchers with this information. So here are some of examples of the different types of research that has been published using data within the consortium. In addition to having this opportunity to do an intermember project where you propose your own research using the available information, there are these research interest groups within the consortium where it is really intended to foster discussion and collaboration amongst researchers with a specific area of interest. So at present, we have a sarcoma research interest group, which is targeted at evaluating that very broad landscape. Sarcoma is so diverse. There's a research interest group on inherited risk. So a number of our genetic counselors are involved in this research interest group. There's an immuno-oncology research interest group, and they are getting ready to do some landscape papers in that area, I believe. Then we have this newly formed GU research interest group. I'm not going to butcher that word for you all. And then we have this ORIEN consortium, which is going to be a new microbiome research interest group that will be forming here in the coming months, where they're really looking at the tumor microenvironment and using some very advanced processing techniques for the transcriptomic data to evaluate the microbiome of the tumor using the existing RNA sequencing data. Within these research interest groups, there is collaboration on research. So each of these groups is working on developing papers, and they can either be single investigator-led projects or group-led projects. So here are some examples of different proposals using this information within the consortiums, or within the research interest groups, excuse me. So I'm sure many of you have come today with a question like, that's lovely. How do I use it? What are the steps to gain access to that valuable resource? So here at IU, in order to use the data and the samples that we have collected locally, we have this kind of three-step process where we would reach out to Pam Rocky, who is our sample acquisition manager, and myself to evaluate the feasibility of your project and establish a cohort of interest. And from there, we invite you to submit a formal request for the samples or data that you plan to use for your project. And then that will go through our standard request process where we send it to a utilization committee for review and approval. And once that review or approval is obtained, our team will work on aggregating your biospecimens, aggregating your data. It is, again, important to note that there's multiple ways to engage with our services. We offer both retrospective sample offerings and prospective sample offerings. It's really dependent on what you need for your research. The majority of our users are engaging with this in a wet lab setting, but we do have a growing number of users that are engaging us for a dry lab research. And then the highlight of the day, requesting ORIEN avatar data across the consortium. In order to access this data, you will need to reach out to myself or Heather Penwell, whose email I provided earlier, and we will support you in submitting a request to the consortium. We have some templated paperwork that we support you in filling out and sending to all of the appropriate parties. So the first group that we would engage with is Aster Insights in order to kind of work on solidifying the conditional criteria for your research. Within that CBio portal instance, there is a great ability to query on what is there, but some questions just can't be answered in that space, and we need to reach out to the data sciences team with Aster Insights for further support. From there, we would generate a non-human subjects IRB submission. Some institutions require that in their review process, and others don't. For us as an institution, if we are engaging in non-human subject research, we do not require an IRB approval. So on that previous slide, asking about biospecimens, if we're distributing those in a de-identified manner, that does not require IRB approval because it is non-human subjects. Once your project within the consortium is evaluated, you'll have the opportunity to present to the ORIEN Scientific Committee, which is a group of really dedicated scientists to put forth your ideas and have the opportunity for feedback about your project plan. After that, there is a network across the consortium for engagement. Because of the way that the network is structured, each participating institution maintains ownership of their data. So they have the ability to choose to contribute it or not contribute it to any given project. And some sites even go as far as to select, they will opt into a project, but there may be specific patients that they elect to withdraw from your specific project because they have a competing demand. Then we go to this next kind of middle step, which is site participation assessment. Each institution will have that ability to opt in, opt out. They may need to submit regulatory documents and do their back-end processing to support your research. Sometimes that can happen quickly, and other times it takes a little more time. Each institution within this consortium manages these proposal processes slightly differently. And from once the point, all of your institutions have opted in, Aster Insights will identify your eligible cohorts of patients, and they will send it to each site for their patient approvals. Once that is obtained, Aster Insights will aggregate your patient cohort from a data standpoint, and then we'll push that data into a DNA Nexus folder for you to download and analyze. It's important to note that the timeline for this process is not the same as TCGA. TCGA turnaround time is typically around 2 weeks, whereas this consortium takes about 4 months. It may be slightly more, it may be slightly less. The one caveat to that is that if you propose a project within a research interest group, the timeline is significantly shortened to approximately 2 weeks. Because every institution has agreed to participate in those research interest groups. I think that is all that I have for you today. I really appreciate the opportunity to talk with you, and I invite any and all of you to ask questions about how this may work for your research.
Great. If anyone has a question, you can unmute yourself. I'll start just by asking a little bit about the research interest groups. So how would someone get connected with one of those research interest groups?
So if you want to reach out to Heather Penwell or myself, we can support you in getting connected with the monthly meetings for these groups. We can also connect you with the CBio portal access so that you can really look at the patient information that's available.
And if your project, let's say there was significant overlap with what you were wanting to do and what maybe someone else had already proposed, would that be a consideration then?
So in the rare event that happens within the consortium, they really try to encourage collaboration amongst the 2 lead investigators in order to streamline the process and benefit from everybody's unique and valuable input.
Are there other questions anyone would like to ask? can also ask, is there any cost involved to the investigator?
So for use of our ORIEN avatar data, at this point, we do not have a cost requirement for that. For the use of our biospecimens that are generated through our core, there is a cost associated with that. And you can see our fee schedule online. We do have a multi-structured fee schedule. Some of it are for our cancer center members. There is some subsidy through our CCSG grant. And then we have a bracket for our IU investigators, which would include Notre Dame and Purdue. And then we have an external vendor cost. So if you are a Notre Dame or Purdue researcher and you take a peek at our fee schedule, please know that you would be paying the IU rate unless you are a cancer center member.
There's a question in the chat. Are the research interest groups located at IU or ORIEN?
So the research interest groups are located within the consortium. They're hosted by Aster Insights and they're online. So they're all by Zoom or Teams meetings. They meet on a monthly basis to discuss plans for projects and areas of growing insight in those given areas.
And so they're made up of people from not necessarily the other cancer centers, but could be from any university that wants to use the samples.
So typically, the people that are involved in the consortium are members of those institutions that are engaged in delivering patients, connecting patients to the consortium. Through our cancer center collaboration between Notre Dame and Purdue, there may be opportunities there as well. The question in the chat is, can you please go over what information can be made available for the different cohorts? Is that a fair assessment of that question? So within our total cancer care protocol, locally, there's access to any of the biospecimens that are collected in that group of participants. We are really dedicated to addressing cancer specifically. So most of the patients that we approach are either individuals who are at risk of developing cancer, either due to like a genetic alteration or individuals who are currently affected by cancer. But we do engage a very small cohort of patients that are healthy comparators who may be undergoing a prophylactic procedure, such as a hysterectomy or something of that sort. For these patients, we have the ability to annotate data specifically to investigator request. We also pull a cancer registry report for these individuals locally. So we have the cancer registry information, which is sort of structured and sort of not. They have a lot of free text fields within that. And this may be what the question was really targeted at, which is ORIEN Avatar. And for these individuals, ORIEN Avatar occurs across that consortium. There are about 25,000 people in this program, and they have the longitudinal clinical data, the germline and tumor DNA, whole exome sequencing, and then the tumor RNA sequencing. Here on
Emily, do all of the samples have the sequencing done?
Yes, all 24,000 of them. Yeah. There are, as one would expect, always fallout in terms of not everything meets QC thresholds for genomic sequencing data, but all the individuals that are included in Avatar should have a germline sequencing and a tumor sequencing. There are rare instances where a patient will only have RNA sequencing or only DNA sequencing, but on the whole, people have both. And as I mentioned earlier, on that left-hand side in black is the consortium numbers, which really is best intended to facilitate dry lab research. And the numbers on the right are for our local contributions, and those may or may not be able to contribute to wet lab research also. We have engaged with some of our investigators here locally to do some really interesting instances. So we've worked with our pancreatic cancer working group to build a TMA using patients that are included in the Avatar project. We have over 100 patients that are in the Avatar project that are also present on that TMA. So our researchers have the ability to double down on that sequencing data and broad tissue data.
Emily, this is Anna Maria. Can you clarify on this slide, on the Avatar slide, where we've got the black numbers, so the total and not necessarily the ones that we have. In the blue, we have everything. In the blue, we have the genomics, we have the clinical data, and we actually have the specimens that are available for use. Now, in the black, which you said was available for mainly dry lab, do we have, I know we do not have access to specimens on those patients that aren't physically at IU, but do we have clinical data on those patients that's available to us?
Yes, the individuals on the left-hand side in black should have that longitudinal clinical and genomic data available for research.
So you could either do some interesting correlative dry lab research, or you could at least explore that to sort out any questions that may occur to you to actually propose a project across centers. Is that correct?
So you would need to propose a project before you could do any abstracts or publications using the information generated from those patients.
There are some questions in the chat.
Yes, the first question is, what about immune cell profiling? Is there any data for that?
I don't know the answer to that. I'm going to say at this time, I don't believe there is. I am not very familiar, honestly, with immune cell profiling and all that it entails. So I would need to get back to you on a more clear answer. You know, that's not a great answer, but it's honest.
Could you post put the website address into the chat box?
Yes, we are a shared facility within the Simon Cancer Center. Our global banking operations facilitate banking in 3 different ways. This is more our central tissue procurement function. The request form that I highlighted in that middle bucket for our local practices can be accessed here. Locally, our standard operating procedure from patients who are affected by cancer is to collect tumor tissue and an EDTA2, which we process into whole blood, buffy coat, and plasma, and we store that for banking. We have historically engaged with patients that have hematologic malignancies. We've been out of that space for quite a while, but we are planning to re-enter that clinic space starting next month. So from those individuals, we have plans to start collecting bone marrow aspirates and blood samples to be processed in a variety of ways. We also have the ability to tailor our tissue and specimen requests to individual investigator needs. So some of that is that conversation between Pam and myself in order to evaluate, what do you really need to be successful in doing your research, and how can we help facilitate that by creating that link between our patient and your bench.
And that, I want to add, that's the value of the huge number. So that's the value of everyone that we are consenting to the total cancer care program, or for that matter, pretty much every new patient that comes into the cancer center is our goal. So the beauty of that is they've consented to having their specimens available regardless of what their specimens or data is. We don't have to reconsent them, and that means you don't need to consent them if you're doing work, as long as it's de-identified. And secondly, you can look at their clinical profiles. They've all agreed to be recontacted. So you can look at their clinical profiles, see what tumor tissue is available for them. And if you have a question for which we do not have the specimen, an appropriate request can be put in, and we may be able to, well, we would contact them, but they may agree to give you whatever specimen you need that we don't already have. So there is a value in this, in the data beyond the avatar data, which has been genotyped, et cetera.
Yes. As Anna Maria mentioned, we are institutionally focused on engaging with a broader number of people who are affected by cancer. And we have been, over the last couple of years, putting in place team members so that we can engage in universal consent across all of our new patients to the cancer center. So we have started collecting samples from specific cohorts of patients within the clinic space, in addition to the surgical space. And we are engaging in some very new biopsy material collections for specific investigators' interest. Those are all during procedures that would be considered standard of care. They are not outside of the standard of care, as our protocol is a minimal risk protocol.
Another question in the chat was if you could clarify the kinds of biospecimens are available and if the samples are fixed or frozen samples.
So our practice as it stands right now is, if we are banking materials and there is sufficient material, we will split that between a fixed and a frozen sample. But if investigators have specific areas of interest or preservation methods, through conversations with Pam and myself, we can make sure that those happen for you. Recently, we began doing a new OCT embedded frozen block for one investigator. We do have the ability to cryopreserve materials. We use specific SOPs, but that is, those special processing methods are really on an investigator specific basis.
I'd like to thank everyone for coming, and thank you, Emily, for a very nice presentation. And everyone have a great rest of your day.
Thank you all so much.